it's about 12:02. So why don't we go ahead and get started? Our speaker today is Jeff Henderson, and he is a GG alum. What year did you graduate?
I graduated in 2018, so not that long ago.
2018. All right. And your degree was in environmental geoscience, environmental science, correct?
Correct. I did have a minor in geology.
Minor in geology. Perfect. Okay. So yeah, let's welcome Jeff, and he's going to be talking about his master's work at the University of Wisconsin. So thanks for joining us from Yellowstone.
Yeah, I'll touch more on that. Thanks everyone for joining. I can only see six of you all at a time. So for everybody in the back, thanks for joining. It's my pleasure. As Julie said, my name is Jeff Henderson. I graduated in 2018, and I'm currently a graduate student at the University of Wisconsin. I'm at their La Crosse campus, and I'll talk more about my experience in the next slide. But what I'm studying is aquatic biology. Specifically, I am looking at habitat restoration in aquatic ecosystems. Let me figure out how to change slide. All right, here we go. Like I said, I go to school at the University of Wisconsin in La Crosse. It's located in La Crosse, Wisconsin, which is on the Minnesota and Wisconsin border, right along the beautiful upper Mississippi River. If you're not familiar with that area, give it a Google search. It's a unique area right in the heart of the driftless region that was changed by glaciers over the several thousands of hundreds of years ago. So due to COVID and kind of everything that's going on, my research kind of got postponed a little bit. I should have been done this past August, but we're looking at a December thesis defense time. So right now I'm currently working on my data analysis and writing in my free time. Some of my experiences in grad school, I think a lot of you are probably undergrads potentially looking into grad school. Some of my experiences, journals and different publications, you read a lot of literature, which is interesting. You get to learn a lot outside of class. I've done a lot of research proposals and grant writing. That's something that you'll become really sufficient at when you go to grad school. So what's unique about my research or my thesis work is that it is fully funded, and that came from a lot of grant writing and a lot of proposals. What's unique as well in grad school, depending on what you go for and stuff like that, partnerships and collaborators. I'll get a little bit more into that when I talk about my research specifically. But yes, I have a bunch of different partnerships with a lot of different agencies and organizations, specifically the USGS, U.S. Fish and Wildlife, Army Corps Engineers. I collaborate with a lot of state agencies and organizations like Wisconsin DNR, Minnesota's DNR, Illinois DNR as well. And then some of the best part of grad school is the field work you get to complete. You learn a plethora of field skills and lab skills. So the fun part is being out in the field, but then you have to go back into the lab, analyze your samples, and stuff like that. And then what I want to touch on too, which I had the unique opportunity as being a teaching assistant and also a graduate assistant at the university. So I was at Slippery Rock for environmental science. I transitioned into the ecology and biology field for grad school. So I was actually a biology lab assistant. It sounds like I assisted somebody, but I independently taught my own biology lab section. So each semester I had anywhere from 3 to 4 lab classes, and I taught them each week. And that was an awesome opportunity. I learned a lot about biology, and I also learned a lot about teaching. So yeah, if you have any questions, there'll be plenty of time at the end. I'll just throw that out there now. You can ask me about preparation for grad school and stuff like that. Right now, I'm currently living out of a conversion van with my girlfriend. We're currently set up right outside of Yellowstone National Park in the town of West Yellowstone, if you're familiar with that. That's our 20-year-old van. hence the backdrop. I'm currently in it, and we're bumming Wi-Fi off the community park. So shout out Smith for the inspiration of van life. Now I'll hop into my research, my thesis research specifically, which is titled "The Role of Large Wood in the Restoration of Habitat in the Upper Mississippi River." Basically, my goal or objective for everybody from this talk is to gain a better understanding of how we can use large wood, which is a naturally found material, to improve habitat in large rivers and potentially other aquatic ecosystems as well. I always start with this, just some of my acknowledgements. Probably nobody's here. I sent the link to some friends, but I don't know if anybody joined. But I just want to give a shout out to everybody that was involved in this study. As you can see, there's a bunch of different people across a variety of different organizations. Specifically my research committee, my sources of funding. I have several sources, so shout out to all them, all my collaborators at these different agencies, and then my field and lab assistants, which were many of my colleagues that helped out with our field work. So to start off, if this is an unfamiliar topic for you, large wood, what is it? It's naturally organic wood that is greater than 10 centimeters in diameter and 1 meter in length. So your typical log basically is classified as large wood. A lot of different literature mentions or acknowledges large wood with different terms. So if you hear me talk about large woody debris or LWD, sometimes you hear coarse woody debris, or a lot of times when wood is negatively perceived, it's called a snag. But there's a lot of different terminology for large wood. And there is also terrestrial large wood and then aquatic large wood. And I'm more focused on the aquatic large wood. So I'm just going to refer to it as large wood. But what does it consist of? It consists of basically any part of a tree. You have branches, you have logs, stumps, you have entire trees that potentially can enter these aquatic ecosystems. That's all considered large woody debris. Now, how does it enter these aquatic ecosystems? You can see by that illustration on the right. It comes from a variety of different habitats, and these habitats are usually within the floodplain. So it can be these terrestrial riparian corridors where wood or where trees fall down and can enter the river. A lot of it comes from upstream environments. So we think about the Upper Mississippi River, which is my study area. There's plenty of tributaries that enter that river, and wood can come from these tributaries. So not only does it come locally around the aquatic ecosystem, but it can come from other ecosystems and be transported downstream. How does the wood enter? Usually it's from different climatic events. You know, if we have a severe flood where the water level rises, we can lift pieces of wood, and that can be transported into the river. Sometimes wind is all it's needed. Wind will blow down a tree right into the river, and the force of the water can actually move the tree or the wood around as well. Now, it's important, before we get into how wood can be used for restoration, the history of wood. Wood in rivers specifically, there has been a great loss over time. The physical removal of wood, which still goes on today, is the main way wood is decreasing, or the abundance of wood is decreasing in rivers. So it began in the early 17th century, and then it accelerated in the 19th century with the development of the steamboat and the timber industry. And the reason why we removed wood was because it created potential hazards and difficulty with navigation and infrastructure downstream. So if you can think about this time when the Industrial Revolution was going on, and we had boats traveling almost every major river in the United States, any piece of wood that was floating by could cause damage. We were doing a lot of bridge building at this time as well, and wood can pile up around bridge pillars and potentially damage them. So we physically removed wood for about 300 years, and it continues to occur today. Deforestation to the watersheds is also another reason why we're losing the amount of wood in rivers. The floodplain surrounding the upper Mississippi River notably is becoming more urbanized and developed. And basically when that happens, we're clear cutting our floodplain forest. I have some colleagues that study the floodplain forest, and it's pretty surprising and alarming the rate that they're decreasing and not being able to regenerate. And then last one, urbanization as a whole. You can see that bottom image, the upper Mississippi River has many dams, and I'll talk more about the lock and dam system later on. But these dams disconnect the flow regime of the river. It stops the movement of wood. So basically wood gets trapped behind these dams and they have to physically remove it. So wood is where it used to be deposited is no longer able to be deposited on these sandbars, on these island shorelines. And that's all from urbanization. We have created locks too, not just dams. And basically any infrastructure in the river channel will impact the movement and transport of large wood. So more into some of the interesting stuff. I just want to point out that there are two main components of large wood. We have a geomorphological influence and a biological influence. And my research focuses more on the biological influence, but I'll touch on the geomorphological influence. So geomorphology is the shape and structure of the river channel. When we have large wood present in a river, it can be deposited in certain areas, which can create sandbars, it can create scores within the river, and it can create log jams and stuff like that. And that all deflects and changes the flow direction, the flow magnitude, and the velocity of the water, which can create it can enhance erosion in certain areas, but it also can prevent erosion. So there's a whole bunch of important components of large wood and how it interacts with an ecosystem. But my main focus is on the biological or the ecological influence of large wood. And I just have some bullet points and I'll run through them real quick. And this is like where I was talking about, where you'll read a lot of primary literature. There's lots of studies on wood in aquatic ecosystems and how they or how organisms benefit from them. So just to start off, wood is a physical habitat for organisms. As you can see in that illustration, if you're familiar with the area of La Crosse, there's many bluffs. So this illustration is kind of like the upper Mississippi River with the bluffs in the background. But it's a physical habitat. It allows organisms to seek shelter and refuge. It's a natural substrate for periphyton. So periphyton, which is one of the main components of my research study, is this mixture. It's kind of like if you think about a blender of all different organisms. It includes algae, it includes macroinvertebrates, it includes microbes, fungi, detritus, the list goes on. So periphyton is basically it's that green slimy stuff that you see on rocks and any wood in a river if you've been near one. So it acts as a place for the periphyton to grow on and develop. And then it also attracts upper trophic level organisms, which is kind of where I'm drawing a lot of my conclusions for my research from. If we look at a food web, the bottom of the food web is our primary production and our secondary production, which are the macroinvertebrates and the algae that grows on the wood. Well, this attracts other organisms, which are a food source for higher level, bigger organisms. So if you think about bald eagles, blue herons, catfish, largemouth bass, pike, these are all upper trophic level organisms that actually benefit from large wood. Some of them benefit directly and some of them benefit indirectly. It's also unique is that studies have linked large wood as a site for plant propagules to grow on. So plant propagule is just a plant seed. And when birds and other organisms that eat seeds use large woody debris or large wood as a perch, in a not so scientific word, their poop has the seeds in it, and this can grow new plants. So if you look at that image by the turtles on the left, you'll see some silver maple seedlings actually growing on wood. And this is important as our floodplain is decreasing. Potentially wood has the ability to house and protect plant populations in downstream environments. And then the also benefit that lacks a lot of scientific research is the nutrient cycling that comes with large woody debris. When wood decomposes, there's a lot of carbon and nitrogen that enters the ecosystem. And when we're talking about a large river like the upper Mississippi River, that is a lot of carbon and nitrogen that potentially can enter the ecosystem. Just a fun fact, when I began this research project, there was a lot of interest in the carbon and nitrogen fluxes due to large wood, but I'll be honest, I was never a chem person, so I steered away from that route. But it is something super interesting and something that is worth noting. So we know that there's all these benefits of wood, and wood has commonly been used in restoration projects all over the world. Specifically in Europe, there's a lot of stream restoration projects that are utilizing wood as a soft material for engineering or preventing erosion in comparison to a hard material, which would be your concrete or rock. So wood is used for habitat improvement or rehabilitation. It's used for bank stabilization, erosion control, and like in the upper Mississippi River, we use wood to protect, create our man-made islands from wind and water activity. Most of these restoration efforts focus on streams and small rivers though. So if you're a trout fisherman or spend a lot of time around streams, you'll notice that we put a lot of wood structures in for habitat. But this brings the knowledge gap. What about large rivers? In a lot of literature that I've analyzed and reviewed, a lot of them talk about large woody debris or large wood in smaller rivers and smaller streams. But there's a big lack of knowledge about large wood in large rivers. Specifically, the knowledge in understanding the restoration potential and the ecology of large wood in large rivers is almost non-existent. So this kind of makes my research unique. We're talking about one of the largest rivers in or in the United States, the Mississippi River, and we're talking about the upper Mississippi River, where we still have a pristine ecosystem, as you can see in that picture. We have main navigation channels, we have secondary channels, and we have backwater habitat. So it's a really unique environment, and potentially we can maximize the wood or the benefits of wood that is already there. So I'm going to jump into some of like the more specifics about my research project. And my study area is considered, or my study area is the Lower Pool 8 Islands Restoration Project. And if you look on the left, you can see just a generic map. You'll see UWL, that's the campus I went to, right in La Crosse, Wisconsin. On the left side of the river is Minnesota, on the right side is Wisconsin. And then the two other arrows, just to point out, we have so many dams, lock and dams on the river. So above Pool 8, you have lock and dam number 7. Above that lock, or above that dam, you have Pool 7. So I'm specifically looking at Pool 8, which is located between lock and dam number 7 and lock and dam number 8. And within that pool, I'm looking at the lower end of that, where this Pool 8 Islands Restoration Project occurred. So as you can see in that first image, in 1890, that's what the Mississippi River looked like before there were any dams. And just to point out, this is just Pool 8, what we're looking at. So that is just an aerial image or rendering of Pool 8 before Lock and Dam 7 or Lock and Dam 8 were built. And as you can see, there's many different channels. And the Mississippi River was once filled with rapids and small slough areas. You could actually cross from one side to the other side and wouldn't be below your knee deep. So it's a truly unique ecosystem at one time. In 1937, you can see that we built Lock and Dam number 8 at the bottom of Pool 8. Over the next 50 years, that water started to pile up, and over that 50 years, we lost 90% of our island acreage. So you can see around 1983 how all those islands basically were non-existent. And it's not because they were underwater necessarily, it's because when we have a larger body of water, it's more exposed to wind activity. With more wind, we have more waves. So these islands were actually being eroded. They weren't just simply being flooded, which is unique. And if we think about all these islands, and we know what the history of the upper Mississippi River Basin, all of this habitat was specifically or was specific areas for waterfowl migration, for fish migration. Even animals could cross the Mississippi River and utilize these islands. And we basically got rid of it all by these lock and dams. So in 1989, they developed this three-phase project known as the Lower Pool 8 Islands Restoration Project. And in 3 phases, they were going to restore these islands. So they actually dredged a lot of the river, creating these sand islands. And phase 1 was completed in 1989. Phase 2 was completed in 2000. And phase 3 was actually completed most recently in 2012. And you can see that we created some of these islands. These are all man-made. However, we're nowhere near where it used to be in the 1890s. So knowing that and using that as my study area, I had 2 objectives for my project. My first objective is, I call it the Natural Wood on Island Surveys. And this was basically to determine what the abundance of large wood, so how much large wood is on these islands, and basically what environmental factors or what factors influence the movement, the loss, the recruitment of this wood. So trying to just going over it broadly without going too specifically, this started in August 2019. So it was the fall of 2019, and we repeated it this spring or this summer in July 2020. So basically what we did was we picked 6 sites, basically 6 islands, and we went to these islands. And on each island, we set up a 60-meter transect. So we flagged out 60 meters of shoreline, and then we walked 10 meters into or onto that shoreline. And we basically had our own transect right there. We started at one end and we walked the transect, and any piece of large wood, so any piece of wood that was larger than 10 centimeters in diameter and 1 meter in length, we counted it, we measured it, we categorized it. When I say categorize, we described it. Did it have bark on it? Was it bare? Was it rotted? Did it have leaves? Did it have limbs? Stuff like that. And then we also tagged it, and you can see how we tagged it. We used an aluminum forestry tag with a number on it, and we used the GPS to record the location. And that was completed in fall 2019. And then we went out again this summer, and we tried to relocate our pieces of wood. If we found new wood, we re-tagged that and we categorized that. If we couldn't find the wood, then we counted that as a loss. And I'll talk a little bit more about that later on. But basically what we were interested in, how much wood is there, is new wood being recruited? So is new wood being deposited on these islands? Is the wood that was already there being removed? And basically develop some kind of turnover budget or turnover rate of how much wood is moving through the system. The second objective of this study, which is the most interesting and captivates a lot of people's ideas, I call it our wood substrate samplers, and basically it's an experimental log. And the goal of this objective was to determine what factors influence periphyton and macroinvertebrate colonization, abundance, and diversity on large wood. So as you can see on the right is an example of one of these wood substrate samplers. Each sampler had an eye bolt and had 5 slices of green ash wood. The reason why we use green ash wood is because in the upper Mississippi River floodplain forest, we have the invasive emerald ash borer, and that's knocking down a lot of the trees. It's basically killing our green ash, and when a storm rolls through, you know, these dead trees fall over, and those are being added to the river pretty frequently. So it was the green ash wasn't just random. There was some logic behind that. And these samplers were buoyant. You know, they floated in the water column about 1.5 meters in depth, and we deployed them. We had 18 of them, and we deployed these 18 samplers over a hydrologic gradient. And what I mean by that is we picked different areas within the Lower Pool 8 Island project that had high flow velocities, which would be our main navigation channel. And then we picked some that were in backwater habitats, which were low flows. You know, there was more vegetation and the water was a little bit slower and shallower. And then we picked some intermediate areas that we call medium flows, which would be our secondary and tertiary channels that did receive some flow, but not necessarily at the magnitude of our high flow sites. So these were deployed in June 2019. And then every 2 weeks we went out and sampled. So how did we sample? We removed one of these wood slices. I have some pictures on the next slide to show you. But what we were sampling was periphyte and macroinvertebrates. So here's like a time series. You can see the first images of images, all of our wood substrate samplers in lab. And then we went out and we dropped a cinder block with some rope attached to it and tethered that to the buoy. So basically our samplers were stationary in the water column. You can't just go add cinder blocks to a river. I had to get special permits for all this and approvals, if you're curious. And basically every time we went out to sample, we removed one of these wood slices. As you can see, that's my field assistant, Alex. We would remove the macroinvertebrates off a specific quadrant. So all these slices, or all these wood slices, were measured and recorded. So I knew the size and area of every wood slice. So we'd pick off the invertebrates, and then we'd use a stencil. As you can see in the top right image, we would scrape the periphyton off. So we'd use a brush and a stencil, and that stencil had a known area, and we'd brush that periphyton off into a tray, which would go into a sample bottle. So this is unique. There's no literature on this process. This was all developed by me, which I'm proud of. It's a unique sampling method, and I've got a lot of good feedback from this. And just to put it in context, there's a map of my study area. So if we're looking at lower pool 8, a lot of those, or those brown areas, those are all the man-made islands. Those black dots are all my sample sites. And as you can see, they're in a variety of areas throughout the river. And then I included some pictures. You can see the buoy, which was camouflaged. That if you're curious why there's brown and green duct tape, we had to camouflage them because people began messing with them. And also there was a note written on each that since this was funded and supported by different federal agencies, that tampering with these was a federal offense. But that still allowed people, or gave people even more, or more of the idea to mess with them. We did lose some of these samplers throughout our study, throughout our project. But as you can see, there's in a different or a variety of different habitat. Some were surrounded by aquatic vegetation, some were more open, but they were all around where previous large wood was observed. So we didn't randomly just drop them. We wanted to mimic large woody debris to the best of our ability. So a lot of these shorelines, as you can see in the top left image, there's a piece of large woody debris entering the water right there.
Hey, Jeff, this seems like it would have been pretty time consuming. How often were you out on the water?
This summer, so it wasn't this past summer, it was the summer before, about every other day I was out in the water. Sampling, we did sampling every 2 weeks, but to reach every site was a bit difficult, and so it took multiple days. As you can see, our top sampler was Horseshoe Extension, and our bottom one was C4 West or C8 West. Those are just the names of the islands. That is about a hour boat ride. You know, we don't really have fancy boats at the university, but it was about an hour to get up top on a calm day. Any wind caused some problems. So it was super time consuming. I had a good team with me, and we were able to knock it out about in 3-4 days for each round. Yeah, but it was fun. You can't beat being on the river and getting paid to do it. So like I said, I'm currently in the process of data analysis and writing. So I have some findings. You know, nothing is set in stone. I'm still waiting for some tests to come back, but I do want to share some preliminary findings. From our objective 1, which is our natural wood on islands, that's where we are analyzing the abundance, the recruitment, the loss, of wood. Our wood characterization, we found that most of the wood was dominantly unattached. So when I'm saying unattached, I mean it's not anchored, it wasn't part of a log jam. It was just by itself basically, and a lot of it was dry. So how we determined was if it was dry was whether if more than 50% was underwater or above water. We found a lot of it was above water, and most of it had or did not have bark, which is unique. When we repeated these surveys this past summer, a lot of the new wood that we found had bark on it, which may helps indicate that that is a newer added piece of wood into the river. When we talk about abundance, you know, I said we picked 6 different sites. Those 6 different sites all represented different island ages. So if you remember, that first phase was completed in 1989, and that last phase was completed in 2012. We picked sites across that spectrum, but we didn't find any significant differences between the wood abundance or the wood size at these different sites, which is kind of unique. I predicted, or I hypothesized that we would find more wood at these older islands. You know, over time wood accumulates more, but we actually didn't see that at all. And then in terms of recruitment and loss, we saw a positive net change, about 3.2 new pieces of wood at each site. You know, some sites we lost 5 pieces of wood, but we gained 8 pieces of wood. But overall, there was a positive net change in wood. And now what I'm at in terms of this is what characteristics influence this turnover? What factors are allowing pieces of wood to be moved and allowing other pieces of wood to be deposited? And just to kind of share with everybody my thoughts, a lot of it has to do with the hydrological and hydraulics, sorry about that, hydraulics of the river and the water itself. In order to lift wood, you know, you need elevated water, you need some kind of flood, but there also needs to be power behind that water to actually move it. So I'm looking in GIS right now, and I'm comparing different inundation models with my 6 sites to see if there's some sites were submerged or inundated more than others, to see maybe that the sites that were flooded more frequently or flooded longer actually have a greater turnover than sites that didn't. So I'm using GIS right now and running all different models to compare and kind of determine why we're seeing this positive net change. And then back to the more interesting stuff with these wood substrate samplers, which is objective 2, is develop an idea of how colonization from these primary producers and these secondary consumers develop on wood. We did find some unique trends, and like I said, I have a graph in the next slide that shows some of this, but these are just preliminary findings that I've observed. Every time we'd pick invertebrates off a wood slice, we place them in sample files. And as you can see in that picture, on the left, we have our low flow sites. You know, that was sampler 9 and sampler 16. And then on the right, we have our high flow sites, which is samplers 10 and 12. And there's many other samplers, but so these are some of the most easily seen or observed patterns. As you can see in our high flow sites, we have a lot more invertebrates than our low flow sites. And there's a different variety of factors, you know, that we can attribute that to, and that's what I'm doing now, is analyzing. But it's super unique to see the difference in low flow and high flow. And I'll talk more about that on the next slide. The 2 images on the top, so basically I had to process every single invertebrate that I collected. There's a metric called biomass, which is basically the mass of an organism in a given area. But I wanted to know the biomass of invertebrates on these wood slices. I wanted to know how many invertebrates were there. So I analyzed every single individual invertebrate. I measured every one. I identified every one. And those are just 2 of the common ones that I found on our wood. The first one is a net spinning caddisfly. Those are unique. They create cases on the wood, basically almost like a cocoon, and they're filter feeders. So as water moves past, you know, they'll filter out the particulate matter, and that's their food source. And then the next one is a flat-headed mayfly, and those are super unique. They have an aerodynamic body morphology or shape, and they're actually more common in higher flow areas. And they they have claws on their legs, and they can attach themselves to basically any substrate in a high flow area. So it's interesting. We're seeing specific invertebrates at these specific locations that matches their adaptations that they have, which is super interesting. We've also found specific invertebrates that are specifically found on wood that we haven't found in years of research in the upper Mississippi River, but we found them on our wood samplers. So basically here's just some graphs, nothing too exciting, but like I said, the main components were periphyte and ash-free dry mass, which is biomass of that periphyte in mixture, and then invertebrate biomass. So if you look at the left, or the bottom left graph, you can see there's the x-axis is our time, the y-axis is our periphyte and biomass, so basically how much periphyton is on this wood given an area. And you can see our different flow velocities, our low flow, medium flow, and our high flows. Well, there's not many or not really a difference between the three different flows, which is unique, but also kind of predictable. But what is unique is if we look at the other graph, that's our invertebrate biomass. So basically how much area on our wood is covered by invertebrates. And you can see that our high flow is greatly significant versus our low and medium flow starting out. And there's a couple different things to pick up on this. You know, initial colonization is more rapid at the higher flows than at the lower intermediate flows, and it's more higher overall. And yeah, basically what I'm doing now is I'm running different statistical analysis. You know, I'm entering different variables and different factors to figure out why are we seeing this. And one of the ideas is potentially that the more flow that we have, you know, the wood is being exposed to more organisms and stuff like that, which is super interesting. So yeah, I talk pretty fast. Just to summarize, there's a lot still going on with my research, but we're nearing the end. You know, I can almost see the rainbow. A lot of it's just writing and stats right now. But large wood is important ecologically. There's a lot of different organisms that benefit from it. Us humans can benefit from it as well. And my research, like what I want to be able to understand is how wood moves through a riverine ecosystem. And if we can better understand that, we can better improve where we're adding wood restoration structures into these restoration projects, so we can improve site selection. Also, if we can analyze primary and secondary production, which are our periphyton and macroinvertebrates, then we can develop some kind of time sequence so we can figure out how they colonize on large wood. We can really improve and benefit our restoration techniques for upper trophic level organisms. So if we know what fish and we know what birds and turtles, we know their diets, and we know what organisms or invertebrates they're eating, if we can better place wood that is more suitable for their diets, you know, we can benefit their populations. So we can improve turtle populations, we can improve fish populations, and even waterfowl, eagle and blue herring populations, which would be awesome. You know, those higher or upper trophic level organisms have a lot of management interest with different agencies. And then overall, for the future, the outlook, is there going to be more or less wood in the upper Mississippi River in the future? It's a great question, and I have some ideas. And basically, if we're thinking about more wood being entered or where this wood is coming from, we got to look at the floodplain forests. You know, that's where our wood originates. Large wood is the linkage between terrestrial environments and aquatic environments. So if we think about the floodplain forests of the upper Mississippi River, we have a lot of silver maples, and these maple stands are reaching their longevity. A lot of them are dying off. You know, a lot of them are falling down. And then we also look at green ash trees. Silver maple and green ash are the two most popular trees found in the floodplain. We have the emerald ash borer, that invasive beetle, which is wiping out our green ash trees. So we have two different trees that are entering at an accelerating pace. And then if we take the idea of climate change, which is something super unique. You know, the Mississippi River that last spring saw one of its highest floods in the past several years. It was almost up 14 feet from what it usually was. So if we're having these stronger, more frequent and more intense inundation events from our spring flooding, you know, we have more erosion. And a lot of these islands and a lot of these shorelines that have our floodplain forests are being eroded away. And that is another way wood can enter. So I predict that we're going to have more wood in the upper Mississippi River in the future. Now, is that a good or bad thing? It all depends on how you look at it. From a restoration standpoint, it's probably a good thing. For the $1 million or $1 billion industry of the shipping channel and the navigation within the river, it's probably a bad thing and is going to cost them money. Basically, this research as in a whole is to better understand the role of large wood in the upper Mississippi River system and other large rivers throughout the United States and potentially other rivers globally. And that's basically all I have. I have some references, awesome publications if you're more interested in this topic. And then any questions we can go on from there. I think
so there's already some questions on the chat. I can read them to you. The first one is, why do you think there is no significant difference in the amount of wood between the old sites and the new sites, unlike what you expected to see?
A main idea, and that's what I'm using these GIS models right now to analyze, is, you know, it didn't really come to me until this past summer when we completed those repeated surveys, is that when we got to some of these islands were higher than the other islands. So elevation of the islands is definitely a factor. You know, the older islands from that phase 1, they built those higher above the water surface than some of these more recent islands. And I just think that the reason why I'm looking at these flood models and these different inundation maps is basically, in order for wood to be deposited on these higher islands, you know, the water is going to have to come up a lot more. And I just don't think that the water is coming up that 17 feet up the bank to deposit that. So yeah, I think that's the main reason why we're not seeing more wood. You think an older island has more mature trees, the forest is a bit more development. So the island itself could be adding trees to it, but we didn't see, or we didn't find that to be the case either. Hopefully that answered the question.
Okay, that was from Lydia. The next one is Dr. Schiappa. You want to ask that, Jan?
Sure. So Jeff, thank you so much for coming and speaking to us. I don't know, I'm pretty pleased that you're doing stuff with invertebrates. I don't know if you remember a conversation we had along the river at a party, and you were talking about, you know, going to grad school and what you were going to do, and we were by the river, and I said, it's all about the microscopic organisms in the river. Do you remember that conversation?
I think I do.
Yeah, well, anyway. So anyway, my question is, do you see a change, like how, do you see a change in the invertebrate population with the seasons?
Yeah, to basically answer your question, I've been looking more broadly at my invertebrates right now, and my plan is to get more specific with the types of invertebrates that we're seeing. So the literature basically states that invertebrate colonization on basically any substrate, whether it's a rock, whether it's wood, any substrate that's in water, is really an initial like 3-week period. And I think that's why we can see a steep increase in colonization right off the bat. But then if you look at that medium flow, if I go back to that graph, you can see that the medium flow actually increased later on. And there are different invertebrates that you know, emerge from their life stages at different times, and at different times of their life stages, they have different habits, and sometimes their habits are to be free floating in the water column. Some of them are they emerge into the terrestrial environment, and some of them stay on substrate longer than others. So I do think you know, I'm still analyzing it right now, but I think there will be some unique patterns that later on in my study, we have more of this invertebrate occurring now than we did at the beginning, if that makes sense.
Yeah.
I basically, when somebody asks me what I study in grad school, I really study aquatic invertebrates. I took 3 different aquatic entomology classes, aquatic, freshwater classes, and really dove into the science behind invertebrates and their feeding guilds and their habits. So they're really unique. We use them as biotic indicators of a lot of different ecosystems.
Yeah, super important. My other question is do they have a preference for wood type? So I don't know if you said what type of wood you're using for your, your system, but do you think, is there a preference? Do they prefer an ash versus an elm versus an oak?
Yeah, that's a great question. Our original idea was for every sampler we're going to make, or for every green ash sampler, we're going to make another sampler that was either maple or another a tree species. But just the logistics behind all that and the money behind creating the samplers, it just was not feasible. There is some literature. A lot of the hardwoods, these invertebrates use like the cracks and the holes and the unevenness of the surface of the wood, and that's where they live. The caddis, they create a web over the cracks. If you get a hardwood that's really hard to infiltrate and there's not many holes, you probably will have less invertebrates. The softer wood that's easier to decompose and has more cracks inside it, you'll see more invertebrates. So again, like tree species, I'm not exactly sure, but like the hardness and the type of wood, there's definitely some differences in invertebrate populations.
Very cool. Thanks, Jeff.
Yeah, thank you.
The next question is, well, there's 2 questions that are similar from Kaylee and Brett. If you guys want to unmute and ask those questions.
So my question was would you say that the higher flowing sites provide more nutrients for these invertebrates to populate on the wood versus the lower flowing sites?
Yeah, so I have 2 ideas regarding why we're seeing more invertebrates overall on our high flow sites versus our low flow sites. When you think of habitat conditions, a high flow site is our main navigation channel. And although the wood samplers are all in the same depth, a higher velocity of water is not really favorable for aquatic vegetation. In a low flow habitat or a low flow site, aquatic vegetation can easily grow and it doesn't have much disturbance. So when we're looking at more invertebrates at our high flow sites, potentially we're seeing more invertebrates because this is the only habitat that's there for them. When we look at our low flow sites, there's different vegetation. There's eelgrass, there's invasive aquatic vegetation, there's milfoil, there's a lot of different aquatic vegetation, and that also provides habitat and a substrate for the invertebrates to live on. So what I'm doing now too is I'm analyzing or I'm running tests to see if vegetation or the presence of vegetation has an influence on invertebrate abundance. So that's one way to look at it. Potentially, like in a high flow site, there's not much habitat, so we dropped in our wood sampler and now they're colonized on it. In the backwater habitat where there's a lot more wood, just naturally found there, and there's a lot more vegetation, potentially the invertebrates aren't seeking out my wood samplers because they already have habitat. And then the other idea is that in a high flow site, we have a more volume of water passing through or passing by these samplers. So you can think about the exposure of different bacteria and fungi, and that kind of builds up as the periphyte, and then maybe that is attracting more invertebrates. In the low flow sites, some of my flow, so I have like measurements of low flows. I use gauges to measure. In some of the backwater areas, we had a flow of 0.1, meters cubes per second. It's not moving fast or not being exposed to different things. So yeah, I haven't pinpointed that exactly. But yeah, hopefully that answer made some sense.
Thank you.
the question I had was kind of like, in addition to the question Kayla had, would oxygenation of the water also play an influence? I mean, with the higher flow sites, you're probably going to have more dissolved oxygen, whereas in the low flow sites with a lot of vegetation, especially maybe pulling oxygen out of the water, that might influence those invertebrates as well.
Yeah, definitely. When we do stream work, at smaller scales, DO is one of the main components that we analyzed. Without a high dissolved oxygen, a lot of these invertebrates are specifically or are favorable to high DO. And if we don't have that, there might not be as much oxygen. But that we didn't, when we sampled, we took water temperature, we took flow velocities, we took several other factors, but we actually did not analyze or take dissolved oxygen, which I think would have been important now that you mentioned that. But I don't know if it would have that great of an effect on a large scale study. I don't think the oxygen or the dissolved oxygen would be significantly different in the main channel versus our backwater, which I could probably get in an argument with that about somebody, but I personally don't think we would see that much of a difference. But that is something to think about as well.
I hadn't meant to put you out there or anything. Or
No, it's good. It makes the research stronger.
Okay, the next question is from Lydia.
So I just wanted to know, so are you planning on trying to eventually, if you find that it improves the habitats, are you going to add wood to improve those habitats? Because I know you were saying that it depends on how you look at it, whether it's a good thing or a bad thing, that there is probably going to end up being more.
Yeah, you can look at it. So I didn't really go into much with this presentation, when I'm actually talking more specifically about my methodology. The study area, large wood, has been added to a lot of these restoration projects. So the main restoration project is building these islands to create habitat. But we're adding to the shoreline of these islands, we're adding pieces of wood. So there's this program, it's called the Upper Mississippi River Restoration Program, and it's funded through all these different agencies, federal and state. And they spend thousands and upward of millions of dollars on transporting wood from barges or on barges out to these islands, and then using large equipment to place these pieces of wood on these are at these sites. So we're already doing that. But the problem is there's no monitoring, there's no assessment, there's no follow-up analysis. We're putting wood out there, but is it really doing anything? So basically what I want to know is, maybe instead of just randomly placing wood on the shorelines, should we put it all on one side of the island, that is facing the higher flows? Or do we put it on the back side where it's facing the lower flows? So it's not necessarily that we're going to be cutting down trees and placing them. We're usually already using wood that is dead. That's what we're
that was kind of my next question. I was wondering, where does the wood come from? Is it just stuff from around the area? So yeah, that kind of led into what I was going to say.
Yeah, exactly. So all the wood is already down. We're not destroying habitat. But yeah, if we're having more wood that's in the river in the future, instead of removing it, potentially we can drag it to the nearest shoreline, and that can create habitat. Is basically what I'm thinking.
Thank you.
Yep, thank you.
The next question is from Jessica, and it's definitely changing gears, which is fine.
So I was wondering if COVID-19 affected your study at all?
Yeah, basically, yeah, it did, and I think it affected almost everybody, no matter what university you're at. Yeah, what was it, right about March, when after winter break, when I was in lab, I was analyzing all my invertebrates, I was running samples, Paraphyte and Ashford dry mass is super unique. I also did paraphyte and chlorophyll. Basically, campus shut down till June. So all my samples were basically, I basically couldn't go into lab. I couldn't analyze anything. So that really put me several months back. And then basically, once I was finally allowed into lab, you know, I have my own research lab and nobody else was allowed in it. You had to wear masks and all that. I really was able to start cruising on analyzing my samples, but it really was prolonged a couple months. And then also I had a lot of field help. And when we did those repeated surveys in July or in June, where we went back out to these islands, you know, I had to do those with just one other person, and those took long as well, and were definitely more difficulty. You know, the policy was that you could only have 2 people per boat, and everybody had to wear masks and stuff like that. And you're talking about 90-degree days, you know, on floodplain forest, walking through reed canary grass and stuff like that. It was not fun. So in terms of how it really affected, it really just pushed back a lot of data analysis. You know, I said at the beginning I was supposed to be done and defending my thesis, you know, this month and back in August, but everything kind of got pushed back. So wrapping things up, and I'll have everything done this December. But it sounds bad, but I have a lot of other colleagues, you know, that had samples in lab that are time sensitive. You know, my invertebrates were stored in ethanol, my periphyton was frozen, you know, and those could have been indefinitely frozen and stored. So I didn't really have to restart anything like some of my friends had or had to, but it definitely just pushed back things.
Okay, there's another question here from Rob.
Oh yeah, I was just wondering, like, you know, I do some traveling and camping and things, and so whenever you're transporting wood to deposit on the riverbanks or wherever you're putting them, like, do you take into consideration like any non-native species of insects or invertebrates that might be tagging along with that wood?
Yeah, definitely. So it's really unique. You know, I said we use green ash, and the ash that we obtained to make our samplers, I had to get a permit from, what was it? Minnesota's Department of Agriculture or something like that. I had to get a permit to actually obtain that wood because that wood was once infiltrated by that non-native or that invasive species of emerald ash borer. So how I obtained my wood to make those samplers, I actually went out with foresters from the Army Corps of Engineers, and we went into a forest in Minnesota, and we cut down 2 trees that were already dead because of emerald ash borer. Well, if you look at that, if you remember that map, the border between Minnesota and Wisconsin basically splits the river. To transport green ash wood across state lines, you have to quarantine that wood, and you also have to receive permits. So basically, although I was working in the same area, you know, there's a lot of security and safety measures, so we don't transport more invasive species. Although we're in La Crosse, basically any county along the upper Mississippi River, all of those have been affected by emerald ash borer. So protocols weren't as strict as if I was taking it somewhere else. But that is another thing. Cutting up all the wood, we didn't find any invasive, like the beetles were all gone, obviously. You know, once they kill the tree, they move on to the next one. So it wasn't really a factor. You know, I wasn't too worried about introducing non-native species, specifically invertebrates, when moving wood around or bringing in wood. In terms of invasive aquatic invertebrates, you know, we don't really have that many in the upper Mississippi River, and if there are, you know, they're already there, so I'm not really bringing those in, if that makes sense.
Yeah, that makes total sense, and it's very cool, all the different things that you have to consider in this work.
Definitely, yeah. I thought I would be able to go down, you know, and pick up some logs from the forest and bring those back in the lab, but I had numerous phone calls with county representatives and officials and stuff like that. It's interesting.
All right, we have time for one more question, and that's from Brett. It's a very interesting question for sure.
Yeah, I was actually put up to this, Jeff, and it's probably for Dr. B's benefit that it was done. But does your boss look at your field notebook, Jeff?
Definitely, I got unique notebook, shout out to Dr. B, there's Right in the Rain books, you can drop them into the Mississippi River and everything stays put. Definitely organization is one of the most important things, and you want to do good research, you want to have good data, as I'm sure most of you taking a class with Dr. B has heard. It's important, after a day out in the field, you have your field notebook, and then as soon as you get back, you're going in the lab and you're recording that somewhere else. Keeping good notes is super important. And we had a mix-up. One of our sites was labeled incorrectly. They were actually mixed maps when I was doing data analysis, and it wasn't making sense. And you could go back into your notes and actually figure out what was right. So they're definitely helpful. I have mine with me in the van. I also have a folder full of data sheets and stuff like that. Everything on Right in the Rain paper.
Well, thank you so much for
Thanks, Jeff.
joining us today.
Yeah,
and
thank you.
And giving a talk. I appreciate it. It's good for our students to see alums who are active in the field. Everyone,
I should have put my email in the chat or on the slide. I'll put it in the chat, but if anybody has any questions about grad school or any questions about aquatic restoration or aquatic invertebrates and stuff like that, feel free to send me an email. I'd be more than glad to share some more of the specifics. Once I figure out how to open up chat, I'll add it there.
If you stop sharing, it'll show up. It should show up. You're going to get questions for help with freshwater biomonitoring exams.
Yeah, I saw someone posted that about Dean Nicola. I took a couple classes with Dean Nicola, and he was the one that I discussed grad schools with. And he was actually recommended the University of Wisconsin, their La Crosse campus. So if anybody had him, tell him Jeff Henderson says
I have him right now.
Yeah, tell him I said hi and give him a shout out. Tell him I said thanks. But
Wetlands and aquatic plants. It'd be right up his alley.
Right. Just a reminder that in 2 weeks, October 7th, we'll be here again, and Morena Miles will be speaking. She's also a graduate of the GGE program, and she will be talking about the chronologies of continental ice sheet fluctuations in Antarctica and in Maine. So that should be interesting as well. So thanks again, Jeff. It's good to see you, and enjoy your van life. Keep us all posted.
Yeah, for sure. I appreciate it. And like I said, if anybody has questions, don't hesitate to reach out.
All right.
Grad school or research.
Awesome.
Good luck, Jeff. Take care.
Thank you. Bye.